Thank you so much, Mr. Ferrer. Good morning, the DG. Good morning, my colleagues, and good morning to the press. I'm very happy to be here to share my experience, my research experience with NIMR. My name is Olaoluwa Akinwale. I'm a professor of parasitology and the immediate past Deputy Director General in Nigerian Institute of Medical Research, NIMR. I'm also the Institute's Director of Neglected Tropical Disease Research. I have shared my expertise as a guest lecturer at the University of Cambridge in the United Kingdom, and I have held visiting professor positions at Ajayi Medical University in Taiwan, and the University of Debrecen in Hungary, the University of Science in Malaysia, and I was once an adjunct professor at the Lead City University in Nigeria. I have won numerous competitive grant awards for research, training, and equipment from national and international funding organizations. Through these grants, I've been able to establish 2 research laboratories in NIMR, the Molecular Parasitology Research Laboratory and the Buruli Ulcer Reference Laboratory. I have served as a trainer and a mentor. I have co-supervised 14 PhD and 3 master's students in the field of neglected tropical diseases research. I have also secured some PhD scholarships and postdoc fellowships for some early career researchers. And I've trained several undergraduates in the lab and contributed to some guidelines, WHO guidelines on mentorship to be used in low-resource settings. So in a nutshell, this is my introduction. Thank you very much. So I will give them the floor to introduce themselves. This is Dr. Abraham Ajayi and Dr. Vincent Gyang. So Dr. Ajayi.
Yes, thank you. Good morning to the DG. Ladies and gentlemen, I'm Dr. Ajayi Abraham. I'm a medical microbiologist. I conduct research at the Molecular Epidemiology Laboratory in the biotechnology department here in NIMR. I work on enteric bacterial pathogens, majorly the ones that cause diarrhea. Basically, some of them are neglected. If you look at it, for example, non-typhoidal salmonella strains, my work centers on using molecular techniques in tracking transmission of these pathogens, looking at the genetic materials that aid their persistence in the environment, and also their persistence to cause infection. So, in a nutshell, I do more of molecular biology techniques in actually understanding the mechanism of these pathogens, as it were, to actually help break the transmission link in the food chain. Thank you.
Dr. Gyang, a brief introduction.
Good morning, everyone. I am Dr. Vincent Gyang. I am a parasitologist by training, and I'm also a senior research fellow in the Public Health and Epidemiology Department. My research focuses on the epidemiology of neglected tropical diseases like schistosomiasis, soil-transmitted helminths, Buruli ulcer. And I also have interest on zoonotic diseases like toxocariasis and toxoplasmosis. Thank you.
Thank you very much. So I will just brief us on what diseases are called neglected tropical diseases. The World Health Organization recognizes 21 infectious diseases as neglected tropical diseases, NTDs in short. They are a group of preventable and treatable diseases that are prevalent in the tropical regions of the world. They disproportionately affect populations in resource-limited settings, such as our country, Nigeria, and they are common among poor people, driving them further into poverty. Some of these diseases are prevalent in many parts of Nigeria. Some are found in all the states of the Federation. Some are located in particular regions depending on the weather. The most common among these infections in Nigeria are Buruli ulcer, human African trypanosomiasis known as sleeping sickness. We have leprosy, lymphatic filariasis that is commonly called elephantiasis, then onchocerciasis known as river blindness. We have schistosomiasis, which is blood in urine. We have soil-transmitted helminthiasis, such as roundworms, hookworms, whipworms. Then we have the blinding trachoma. These are major infection, tropical diseases that are common in Nigeria. We also have yaws and the likes. So my team's activities, when I say my team, including Dr. Gyang, our research activities include conducting studies on the distribution of some of these NTDs and understanding their intermediate hosts and vectors that are of public health importance in our country. We employ various parasitological and molecular techniques to investigate the frequency and pattern of these entities, the reasons for their occurrence in the country, and how our findings can be utilized for planning, evaluation, control, and management of the NTDs. We also conduct training and workshops on the application of these techniques to identify the causative agents, that is the parasites or the bacteria, and their intermediate hosts. Currently, we are collaborating with the University of Cambridge in the United Kingdom to study the epidemiology of schistosomiasis, the blood in urine infection. We are using a One Health approach, studying the parasites from the human and animal angle, as well as the environment. So the study is ongoing and is funded by the Cambridge Africa Research Grant. Recently, we conducted some studies. So I'll be talking on schistosomiasis and Buruli ulcer, while Dr. Gyang will be talking on soil-transmitted helminths. Urinary schistosomiasis, as the name implies, is a type of NTD that causes blood in urine. That is, the infected person urinates blood along with in the urine. So the infection impacts millions of people all over the world, especially vulnerable groups such as children and women of childbearing age in sub-Saharan Africa, including Nigeria. We also know that the disease also manifests as a gender-specific infection in some cases. The urogenital schistosomiasis could affect the genital organs of the patients, especially the women. So the gender-specific manifestation of this infection is known as female it's known as female genital schistosomiasis. In Nigeria, there is paucity of data on this gender-specific manifestation of urinary schistosomiasis. So our study now aimed to contribute to the much-needed data so that we can create more awareness in the country. So we set out to screen women of childbearing age in some schistosomiasis endemic communities for the symptoms of this female genital schistosomiasis to determine the prevalence of the infection in the communities. We used microscopy, urine microscopy, clinical examination, pap smear, and photocolposcopy to screen the women using the WHO FGS Pocket Atlas as our guide. The study received funding from grants awarded by the Royal Society of Tropical Medicine and Hygiene and the National Institute for Health Research in the United Kingdom. So our findings, almost half of the women that we screened were infected with urinary schistosomiasis. Mind you, these are women living in schistosomiasis endemic communities, so it's no surprise that half of our participants had this infection. Younger women were significantly more infected. Then the photocolposcopy results showed that about half of the infected women, not half of the participants, half of the infected women had signs that could be considered as female genital schistosomiasis. These signs included graining sandy patches, yellow sandy patches, abnormal blood vessels, and rubbery papules. We worked with clinicians who were trained on colposcopy so that they helped us to interpret the results. And the pap smear results also showed abnormal cervical manifestations. So our recommendations were that more measures needed to be taken to improve the health of women, especially those living in schistosomiasis endemic communities. While all our infected women in the study showed cervical cells that deviated from the normal appearance or structure, it is important to follow up with them to monitor any potential progression of these cells that could lead to malignant lesions. So we strongly recommend that more studies should be conducted with larger sample sizes in these communities and in other communities where this infection is endemic. Those are our findings and recommendations. So we are also looking for more funding to continue this work and to follow up with these women that presented with symptoms of FGS so that we could recommend them for timely intervention in case those cells that we observed that were not really normal as the normal cells, before they could start to misbehave and change to something else that could cause more serious health implication for these women. Then our second disease is Buruli ulcer, which is also common in Nigeria, but not as common as urinary schistosomiasis. The WHO has set a test known as polymerase chain reaction confirmation rate for at least 70% of cases of Buruli ulcer that are clinically diagnosed in the country. These cases should also be confirmed by PCR because microscopy that is commonly used in the field to confirm these cases is not as sensitive or specific as PCR. PCR works with the DNA of the parasite. So whatever the amount of the sample, if the bacteria that causes Buruli ulcer, Mycobacterium multirans, is in the sample of the patient, the microscopy can miss it, but PCR will pick it up. So WHO now recommended that every country reporting cases of Buruli ulcer should subject at least 70% of their cases to this PCR confirmation, because the treatment is for 8 weeks, a daily dosage of antibiotics. You will subject a patient to such. So care must be taken in order not to treat a patient using false positive results. So that is the essence of this PCR. As of 2015, the National Buruli Ulcer Control Program in Nigeria was faced with significant challenge due to the lack of access to in-country PCR confirmation. So we saw this gap, and applied for a grant from WHO TDR to establish this PCR confirmation in NIMR. And as by 2016, we were able to establish the Buruli Ulcer Reference Laboratory for PCR confirmation in the country. And the laboratory is domiciled in NIMR with funding from WHO TDR and support from NIMR. So the laboratory became a founding member of the Skin NTD Laboratory Network in the World Health Organization African region. The network, the Skin NTD Network, has been providing support since 2016 to the lab in terms of reagents and consumables, because PCR test is very expensive. So we have been receiving support from the network. So the network also ensures that all member laboratories in the West African region operates a standard operating procedure so that our results will be reliable, because we are dealing with human samples. And we are talking of antimicrobial resistance, so we don't want to add to it. So the network ensures that all 19 to 20 laboratories in the network spread across West African region mainly. We also have a few members from other regions of Africa where Buruli ulcer is also prevalent. We operate a single standard operating procedure so that our results that we release to the state control program, the national control program at the Federal Ministry of Health, the results will be reliable. And the patients, once the patient is confirmed as having the ulcer, will be immediately placed on treatment and wound management. So the reference laboratory has been helping supporting the national control program in this aspect, so that the gap is already taken care of. And NIMR has, in that wise, helped to ensure timely diagnosis and improved treatment outcomes for patients in the country. Then we looked at our data over time, and our findings since 2016 showed that the Buruli ulcer is not as endemic in Nigeria compared with some other African states. It has low prevalence. And then the results also showed significant difference between males and females in terms of the infection. No significant difference. So there's no difference between how many males are infected or how many females. But we observed that individuals under the age of 50 are more affected than the older age groups. Then our samples have been coming since 2016. We have been receiving samples from about 21 states, but most of the states are from the southern part of the country. And it's not a surprise to us, because this infection is found in places where we have heavy rainfall and humidity. So finding more cases in the southern part of the country is no big surprise. Then our recommendations. Given the results showing low prevalence of Buruli ulcer in Nigeria, we recommend the following: that clinically diagnosed BU cases that we now find to be PCR negative should be further screened for other NTDs like yours that present clinically like Buruli ulcer, so that even if the patients are negative for Buruli ulcer, they can still be treated for the particular NTD infecting the patient. And then we also advocate for increased government sensitization, especially at the grassroots level. Then there should be improved active case searches by health workers, especially in our rural areas, so that more cases will be discovered for timely intervention. In the course of our research, we are faced with some challenges, which is common to all groups: limited funding, and then molecular reagents and consumables are very expensive. We get them from abroad at a very high rate of exchange. By the time we pay for them, we pay VAT, we pay tax, we pay clearance. The tests that we use them now become very expensive. Fortunately, we've been having support from NIMR, thanks to our DG, and from the WHO in Geneva, especially for Buruli ulcer confirmation using PCR, because patients cannot afford the test. So since 2016, NIMR has been conducting this test free of charge. And we have conducted more than 3,000 or 5,000 samples free of charge. Meanwhile, each test is very, very expensive. But fortunately, our institute supports us, and then we get continuous support from WHO in Geneva. So solutions, we intend to explore more grant opportunities, train more early career researchers in grantsmanship, because we cannot rely on government funding alone. Government has been trying funding us, but research involves more, it's capital intensive, so we need more funding. So my intention is to continue to train early career researchers, to mentor them to win grants. So fortunately, we have a foundation in NIMR also that conducts yearly workmanship, workshop on grantsmanship. So we intend to put in more efforts doing this. And then we also want to build local capacity in the area of development of local diagnostic tools, so that even if we have our tools, we have our kits, they will not be as expensive as those that we buy from abroad. Thank you very much, sir. Thank you all for listening.
As part of the work we do in the department, soil transmitted health is what we commonly call worms in the local palace. So these are infections that are widespread in the country, and it mostly affects children due to their level of hygiene. And when they get infected, it leads to anemia, there's vitamin deficiency, and also there will be stunted growth, and it affects their learning ability. So their performance tends to be poor in school. This infection is normally found in rural areas due to the poor hygiene, lack of amenities. But due to the rural-urban migration, it's moving now into the cities. And there's also movement from the urban slums to other parts of different states. Like in our study, in an urban slum here in Lagos, the children were screened, and over 90% were positive with intestinal parasites. And of those 90%, about 50 had multiple infections. Some had 2 worms, some had 3 worms, which exhibited different morbidities in the children. So we now went back to the community and gave them our recommendations. We now advise that those communities should have more regular mass deworming programs, and there should be more education on health hygiene in that area. We also advise that they should have safe drinking water in those communities. And then lastly, we advocated for the complete stoppage of open defecation, since the route of infection is the oral fecal route. That is, we pick it from what you eat. Thank you.
Non-typhoidal salmonella infection, it's quite endemic. It has the burden that is not quite understood. Why? Because surveillance in regard to looking at the infection in the different interfaces, human, animal, and the environment, is quite lacking. In the West, for example, in the US last year, they reported several cases of non-typhoidal salmonella outbreak. On a yearly basis, even this year, February, there was, an outbreak already. The transmission was from war gecko, the gecko, Okay, some people have it as a pet. But you know, here in Nigeria, because of lack of surveillance, we really don't have comprehensive data. The data that we have is from the study by Ajayi, though it was a clinical-based study. So what we're trying to do is to look more into the transmission cycle and how we can break the transmission cycle. Though the infection is self-limiting in healthy people, but there are a category of people, the very young, the aged people, and those with compromised immunity, that when they get infected, you know, it could be severe and can be fatal in some cases. So it is important that we look at the transmission. In Nigeria, from the study we conducted, we found out that healthy food handlers, for example, were carriers of these serovirus. The non-typhoidal strain, okay, let me just explain briefly. Typhoid fever is caused by Salmonella typhi, which everybody gets to know. But the non-typhoidal strains that cause gastroenteritis, people really don't look at it. But it's there. People have this diarrhea which will go after a period of time, and before they even go to the hospital, they self-medicate. It's only when it is severe that you see them come to the hospital. And we've looked at it and found out that there are a kind of evolution in the strain. Some of them are becoming more invasive than what it used to be. So even from animals, we actually isolated some of these organisms from food animals, poultry, sheep, cattle, you know, here in our environment. So the recommendation would be that we should look critically into it. When people are cooking, they should cook their food properly and try to be very hygienic. Because you don't use the knife, for example, you have used in cutting poultry products, meat, to cut vegetables or other things that you will consume without further processing, because cross-contamination happens. And that is the major route where people can, you know, get infected with this pathogen. So more funding is also needed, and we have to beam the light towards the non-typhoidal Salmonella infection around us. Thank you.
Yeah, I was going to say one or two things concerning the presentation. I want to start with schistosomiasis. We've been told it's a parasitic infection. We've been told that poor hygiene, improper water supply, like going to the stream to wash, to urinate, to bath, to swim. If somebody who is infested releases the egg into that kind of environment, another person going into it can pick it up. Of course, once the eggs are released, they go into a specific intermediate host, the snails, they develop inside it, and then when they are released, they get into humans via the skin. When they get through the skin into the body, they go into the blood vessels. And they go through the right side of the heart to the left side, get into the liver, through the portal circulation, a blood network to the liver, and eventually end up in the veins. Those are the vessels that normally carry blood back towards the heart. The veins in the pelvic area, in the lower abdomen, the pelvis. I don't know if I'm communicating. I went through this to draw attention to the female genital schistosomiasis. It's because they are resident within the vessels in that area that they invade the organs within that area, mainly the bladder. You've been told about the fact that you have bloody urine, which we call hematuria. And what happens is that the eggs through the veins penetrate. They're very powerful worms, because to go into the veins is like you are going in the reverse direction. They get into the wall of the urinary bladder. They are dispersed. For some of us, we see these things as beautiful structures under the microscope. And they grow towards the surface lining of the urinary bladder, exposing it. There's bleeding into it. And it is that blood that we see in the urine. Of course, with the ova coming out and being urinated. But apart from the urinary bladder, okay, maybe what I'm still talking about urinary bladder, the schistosoma ova causes an irritation of that inner lining of the urinary bladder. The inner coat of the urinary bladder comprise a particular group of cells, which we refer to as transitional cells. However, because of the irritation produced by the schistosoma, that layer is now replaced by another type of cell that is more resistant. Those are the squamous cells, like the cells of the skin. And that's what progresses on to cancer of the urinary bladder. The classical cancer of the urinary bladder due to schistosomasis is a squamous cell carcinoma. But apart from the urinary bladder, in women in that area as well, you have the uterus, the cervix, the fallopian tubes, the ovaries. And you can have a migration into all those places, even externally through the vaginal wall, producing the ulcers and the grain-like structures, appearance mentioned. If you are not conversant with it, you might miss the diagnosis. Of course, when gynecologists like Professor Ajayi sees something like that, or this abnormal appearance of the cervix, they take a smear or they take a biopsy of the skin, send it to the pathologist in the lab who looks at it under the microscope. I went into this just to explain how we came about the female genital schistosomas. I don't know if you want to add anything to that, sir.
Our home here, what is the federal government doing, or any other agency or organization or whatever, doing to support the research in the area?
Thank you very much for that question. If you could remember, I mentioned that apart from grants from WHO, from TDR, from Cambridge, and some other international organizations, we have support from NIMR. And I even say thank you to DG. NIMR is a government, federal government establishment. So when we NIMR doesn't manufacture or print money, so it's government support. But government alone cannot fund research, because research is a very expensive, capital-intensive venture. And like our DG said, we don't produce materials for sale so that we generate money to pay NEPA and some other things that we use. And to buttress what our DG has just said, I mentioned that all these studies were funded with support from NIMR as well. They're funded with, by international organizations. We competed for these grants, and we are accountable to them. Currently, I told you that the Olaoluwa laboratory is being supported by the network under which we have all the Olaoluwa laboratories in Africa operating with the same SOP. And they've been supporting us since 2016 and all other laboratories with expensive reagents and consumables at no cost to the patients, at no cost to the government. And these expensive reagents are kept, like in our laboratory, we have a freezer that has been on continuous electricity supply since 2008, because that is where we keep our reagents. Since 2008, this freezer has not been without NEPA, without electricity, to show you how sensitive these reagents are. And these reagents are supplied by the network from Geneva and then by American Leprosy Mission from the US and some other organizations with the support of federal government through NIMR. So I really want to support what my DG has said, that you please help us bring it to the limelight, that this is the plight of NIMR. We have freezers, we have reagents that are temperature sensitive. Some should be at -80, some -40, some -20, some at room temperature, very few anyway. The majority of our reagents are kept at particular low temperature. So we also have support of government. Our government is funding us, but research is capital intensive. Government alone cannot fund research. We also need private-public partnership to assist us. We are trying to also source for funding from abroad, but they are highly competitive. They are not just giving us money. You have to compete with your colleagues in the West, US, UK, and some other developed countries that are not facing the challenges we are facing here in this part of the world. We compete with them, and we are all given fair, same field to play. So we are really facing a lot of challenges in terms of funding. But government is also trying, and we receive funding through the government, through NIMR, to support our work as well.
Okay, my question is for you also, we know WHO have a roadmap of 2021 to 2030 to eliminate NTDs.
Certain NTDs.
Is this possible, like in Nigeria? Is it a reality that we can reach this?
Not only in Nigeria that we are facing challenges with meeting that target. Some other, many countries, and mind you, these NTDs are mainly found in developing countries, like Nigeria, Ghana, and some other countries in the tropics. So we are facing the same challenges as how to meet the targets, the roadmap, with well, we are making some efforts, but 2030 is around the corner, and not much has been achieved. So that's what I can see.
Okay, let me ask. Some of us in this room have been at different media dialogue with UNICEF on this entity. I don't know if you have any connection or involvement in what UNICEF is doing around these entities. Okay. And again, I want to know some of these studies you've done, some of the communities where you did them and all that, you didn't mention.
For privacy's sake, because we didn't obtain their consent for this media chats. We will share our publications with you. We mentioned their names there. But for ethical reasons, my colleague is there, who is always fighting us on behalf of the communities. We call him Professor Ethics, to protect the society, especially our study participants. We hardly just disclose their identity without their permission. But we had their permission to publish, and I'm assured you will share our publications with you. So that is one. Then two, you said UNICEF. When I said WHO TDR grants, the grants are a sort of pool of money by some international organizations, including UNICEF, UNDP, WHO, and so on. So they come together, they pool resources together, and then they fund research globally. So indirectly, UNICEF, which is, I think UNICEF is an organ or agency of WHO, if I'm right. So indirectly, they are also funding UNDP. So they are funding research. So it's like indirectly working with us through the grants that we obtain from. But when we say WHO, it's just an abbreviation of a long name comprising of all these organizations that fund.
Yes, sorry, for knowledge purposes, some of these entities, some of us are not able to pronounce them. There's one you're talking about, the female and the male that it affects more. I want to know why. I think it is so weird.
Okay, I think I shared a 6-page document with Mr. Mr. Sam to share with you so that you have a background knowledge of these diseases because of time. So for schistosomiasis, like our DG, I'm even wondering how did DG know so much about? So you'll be coming to our lab to lecture us. I'm so impressed. Thank you, sir. So the schistosomiasis is in 2 parts, just as DG said. We have those ones that belong to the urinary schistosomiasis that cause the urinary, just as the name, and then causes blood in urine. We have those ones that reside in the intestine. I'm just saying this in layman terms, but if I should say it in the technical, it's not intestine per se. DG has gone into some technical aspects, talking about veins and arteries, So we have the ones that intestinal ones, that you get their eggs from the stool of the patient, the feces of the patient, and then you can see blood in the feces caused by this, the passage of the egg through the intestinal wall or something. So when we say more women are affected than men, when you look at domestic chores, for example, in Nigerian homes, we women do more. It is women that will go to the water, the river in this village to wash clothes, to bathe their children. So that's why we are seeing more women suffering from this FGS. Currently, Dr. Gyang has a grant looking into the MGS, male genital schistosomiasis, am I right? Because we also know that both men and women are exposed, because men also go for recreational purposes. I don't think Nigerian men will carry their clothes and go and wash. It's the wives that will do that. But sometimes they go to the river to fish. So fishermen are also exposed because of the nature of their jobs or work. So to fish or to do some other things. So they are also exposed to the infected or contaminated water. But more women in our studies, we discovered that are more affected by this. And just as the DG has highlighted, the passage of the eggs of these parasites through the walls of the genital organs, because the egg has some spines like hooks. As they are moving, they are causing lesions, wounds inside and bleeding. That's why you now see blood. But more children under the age of 19 urinate blood. By the time they transit into adulthood, the adults may not be passing blood in their urine. The infection must have transited into to chronic stage, whereby you think the person is no longer infected. No, the person is infected, but shedding less of the eggs that could cause that blood to be seen in urine. And that is where the danger lies. the egg will be causing some damages, like our DG has explained, inflammation and some other, and they're causing some cells in the genital, in the organs where these eggs are residing, to start misbehaving. That's all, presenting abnormally. I mentioned it, but I just wanted not to create fears in our hearts that they are likely to have cancer of the cervix. No. Studies didn't go that way. But what we found is we saw some abnormal cells. And these women need to be followed up because it may take some time before these abnormal eggs will now start to transform into malignant cells. We just want to play safe so that we don't send wrong signals that women are having cancer, this and that. No. We didn't, studies didn't really go that far.
Okay, if I may still ask my question. Yes, I know that the major focus of the research is to impact the society positively and bring the healthier delivery system to the people. I just want to find out, after your research, is there any follow-up to the community where you conducted this research so that the people or those in authority that should have taken the next line of action to do so, in order to at least give this, the people or the victim or the people, not to prevent them from having a kind of a convenience. Is there any feedback mechanism?
Thank you very much. Our team is not only laboratory parasitologists, we have sociologists, we have those that are trained in health communication. And when we are designing the proposal, we work with program managers, the disease managers from states and from Federal Ministry of Health, because they are the ones that we eventually have our reports and bring the reports to the notice of the policymakers, government, and so on, apart from our publications. And then at the end, we signed a pact while we were signing the grant awards that we will go back to the communities and share the reports with them so that they will know, like these women that we found that half of our participants had this urinary schistosomiasis, and half of those that had also have some abnormal cells that may later in life transform into some malignant cells. So we went back to the communities with clinicians on the team and the program managers and some staff of the health centers there to also share our results with them. And those that we found positive with this infection were treated and were advised also to be followed up for timely intervention.
Creating time to come again today and spend some quality time with us in NIMR. I hope it's been informative enough. I think the next session is going to be by Professor Ajayi. And all I can say is I thank you.
Thank you, sir.